Hey guys, so in this video we are going to study about reproduction in viruses. Okay, so reproduction in viruses are of two main types. One is phagic and the other is pinocytic. Okay, now what is pinocytic reproduction? In this kind of reproduction, the whole virus passes into host cell. Okay, and in phagic reproduction, only nucleoid enters the host cell. Okay, now again we are going to study the phagic reproduction in detail. So phagic reproduction has two subtypes. First being lytic and second is lysogenic. Okay, so we'll study both one by one with the help of diagram. Now let's start with lytic reproduction. So as you can see in the diagram, in lytic cycle, what happens is, so we are first thing is we are going to study the lytic cycle in T4 bacteriophage. Okay, so this is the reproductive cycle of virulent phages. Okay, like T4 bacteriophage. Now what happens is, as you can see this structure A here, what happens is the phage here, this is the phage which attaches itself to the host cell. Say here the host cell is E. coli. Okay, so this is the phage, this is the host E. coli, this is the phage. What it is doing is it is attaching itself to the host cell and it is attaching through the tail fibers. So in the previous video we have seen the structure and diagram of bacteriophages and different kind of viruses. If you are not aware of the structure completely, please go and watch the previous video. So here through the tail fiber the phage is attaching to the E. coli cell and the fiber bends and brings the tip of tail in contact with the host cell wall. So this is the cell wall and the tail is getting in contact with the host cell wall, and then the tip of the tail produces a hole in the bacterial cell. So what it is doing is it is producing a hole here in the bacterial cell, and that is done with the means of enzyme, lysozyme. Okay, so with the means of enzyme lysozyme, the phage is making a hole in the cell of E. coli, and then the tail sheath present here contracts and injects the viral genome. So here this is the viral genome which is injected inside the host cell. Now after entering the host cell, the viral DNA transcribes some mRNA, the messenger RNA. It transcribes mRNA and it forms the enzyme over the host ribosomes. Now, some of these are nucleases. Okay, so basically, this genome is making the enzyme nucleases. Okay, now what nuclease does is it degrades host DNA and mRNA. The host DNA and mRNA degrades, and the transfer RNA and ribosomes remains unaffected. Then the phage DNA and mRNA, what they do is they gets attached to the parent viral DNA functions, and with the help of bacterial nucleotides. Now the host machinery, what it does is it synthesizes proteins and it develops new viruses. So here it is basically transcribing its genetic material, and the phage DNA synthesize proteins, viral lysozymes. So it is synthesizing proteins, viral lysozyme. Then they have internal proteins, they have capsid proteins. So in it is basically what it is doing is it is synthesizing all the materials which is needed to generate individual phages inside the host cell. Now different components, then these are the different components which are transcribed, and they just combine to form new viruses. So here in the picture you can see these are new viruses. Okay, so they combine to form new viruses or phage particles. Then what happens is the host cell ruptures. So here you can see that the host cell ruptures, and the new phages formed comes out of the host cell. Now the host cell ruptures again with the help of enzyme lysozyme. Okay, now there's one term which is eclipse period, which is the period between entry of nucleoid, the viral nucleoid, and bursting so of host cell. So it is basically the period starting from A and completing at E. So this is the entire lytic cycle in bacteriophages. Then we have the lysogenic cycle. Now what is the lysogenic cycle? So basically we'll study the lysogenic cycle in lambda phage. Okay, now here we have two cycles in one basically. So we have the lysogenic cycle here and the lytic cycle as well. Okay, so again the host here is E. coli. Okay, so now here what happens is the phage does not possess a tail fiber, or it does not attaches the tail fiber, but the tail completely comes in contact with the host cell. It drills a hole in the wall and inject the phage DNA into the cell. So it just drills a hole and injects the DNA into the host cell. And what it does is produces a repressor. Okay, so after that it produces a repressor and undergoes a state that is non-virulent state. So in the case of lytic cycle, it enters a virulent state, and in the case of lysogenic cycle, it enters into a non-virulent state. Now with the help of enzyme integrase, the viral genome becomes integrated with the chromosomal DNA. So here as you can see that this is the viral DNA, and here we have the chromosome of the host cell, and with the help of enzyme integrase what it did is it got attached with the host cell chromosome. Okay, so it attaches at a specific site, and at this particular time, the viral genome is known as prophage. What it is called? It is known as prophage. Now, this prophage, along with the, so here you can see that this is the prophage. Now, it along with the bacterial DNA replicates and gets distributed. So, this replicates into many and it gets distributed to the daughter bacteria. So, the here the host cell is completely dividing into daughter cells, and the viral genome is getting distributed in these daughter cells. Okay, so after that, again it goes into the lytic cycle, wherein this prophage is dividing, it is forming various structures of the bacteriophage, it is multiplying, then the entire cell is combining to form various viruses or phage particles, and again the host cell ruptures and the individual bacteriophages comes out of the host cell. So here we have seen the lysogenic cycle and lytic cycle together. Now let's just go back and revise what is the difference between lysogenic cycle and lytic cycle of a virus. So let's see that separately once. Okay, so we'll have the lytic cycle or lytic phase and the lysogenic phase. So it's important to understand the difference between both the phages. Okay, so here the viral genome does not integrate with host cell. So what it does is it actually kills the entire mRNA and DNA of the host cell, and it produces certain proteins to develop the entire structures of the bacteriophage, which combines and forms individual bacteriophages. But in lysogenic cycle, what happens is it integrates the viral genome, integrates with the host DNA, and it is known as prophage. Okay, so this is the first difference. Then in lytic phage, what happens is the host DNA is hydrolyzed. Okay, so it breaks down, but it is not hydrolyzed in the lysogenic phase. Then in the third difference is in the lytic phase, the viral genome. So the viral genome or the viral DNA, it replicates, right? And basically it form copies in the same host cell. So here the host cell is not dividing. Okay, the genome replicates and form different copies. But in lysogenic cycle, what happens is the prophage that is formed, the integrated viral cell and the host DNA, it replicates only once. It replicates only once, and this replication is along with the replication of the host genome, and every single particle is transferred to a daughter cell. Okay, then The fourth difference is here the virus is virulent, and in lysogenic cycle it is non-virulent. Okay, then the host cell in lytic cycle, it undergoes lysis, so it bursts and the virus comes out, right? The bacteriophage comes out. So the host cell undergoes lysis, host cell. But in the case of lysogenic phase, the host cell is not lysed. Okay, it does not breaks down. Then the last difference is that certain number of virus particles are liberated when the host cell becomes lysed. Okay, here virus particles are not liberated. It remains in the daughter cells. Okay, so these are the basic differences between a lytic and a lysogenic phase. So you should remember both the differences mentioned, as it is important from the exam point of view. And here we end with the reproduction in viruses. What we can do is we can go for a quick revision, okay, wherein we'll start with the types of reproduction in viruses. So types of reproduction basically are phagic and pinocytic. Now in pinocytic, what happens is the whole virus passes into the host cell, and in phagic, only nucleoid enters the host cell. Then again, phagic reproduction is of two types. It is either lytic or lysogenic. Now we'll just see lytic cycle first. So basically, it is wherein the phage transfers the genetic material through the tail fibers, wherein it uses a lytic enzyme, that is lysozyme, and then it destroys the host DNA and mRNA. Transfer RNA remains the same, and it transcribes the different structures of the bacterial RNA, wherein we can see there is a tail, there is a head, the genome present. It enters, it transcribes the entire structure of the bacteriophage. It generates various proteins like capsid protein, viral lysozyme, internal proteins, which make up the entire structure of the bacteriophage. Then it forms together the entire structure of bacteriophage. The host cell bursts and the bacteriophages comes out of the host cell. However, the other cycle, that is the lysogenic cycle, is different, wherein the structure of the host cell, what it does is the bacteria gets attached with the entire tail and injects the phage DNA into the cell directly. Once that is done, what happens is it produces a non-virulent state, which is virulent in the case of lytic cycle. So here it represses and makes up a non-virulent state and undergoes reduction. And then with the help of enzyme integrase, the viral genome becomes integrated with the chromosomal DNA of the host cell, and it directly gets distributed. It replicates along with the bacterial cell, and it gets distributed in the daughter cells, and it completes the lysogenic cycle. Once the daughter cells are formed, again the lytic cycle starts, wherein the viral DNA starts dividing and forming various proteins and nucleoids, which attaches, forms the individual bacteriophages. The cell bursts and the bacteriophages comes out of the host cell. So then we studied the entire difference between a lytic and lysogenic phase. The basic differences are that in the lytic phase, the viral genome does not integrate into the host cell, while it integrates with the host DNA, forming a prophage and lysogenic phase. Then in lytic, the host DNA hydrolyzes, and in lysogenic, it does not hydrolyze. Then in lytic phase, we have the viral genome, which replicates and form copies in the same host cell, while in lysogenic phase the prophage replicates but only once, and which is transferred into the daughter cell. Here the virus is virulent, in lysogenic it is non-virulent. Then in lytic it undergoes hydrolysis of the host cell, whereas where in the lysogenic phase the host cell is never hydrolyzed, it is not lysed. Then in the case of lytic, certain virus particles are liberated, but virus particles are not liberated in the case of lysogenic phase. And I hope you understood the entire video. In case of any doubt, please don't forget to comment in the section below. And if you understood the topic completely, please like, rate, and recommend the video. Thank you so much.